This is a production of Cornell University.
Well, thank you guys, and thank you friends for coming. It's amazing our semester has gone by so quickly. And my dissertation focused on a horticultural mineral oil called Civitas. And horticultural mineral oils have been around for a really long time, and you might think, why is this renewed interest in studying horticultural oils? And it's mainly because over the last decade, we've tried to reduce our chemical inputs onto turfgrass systems. And horticultural oils are generally non-toxic and they're low threat to the environment. So companies like Petro-Canada that developed this product are looking at safer alternatives to commercial traditional pesticides to minimize impact and satisfy golfer demand. So kind of a quick seminar outlines of today's seminar. We'll do a brief introduction on spray oil literature, which goes back to the 1700s. We'll talk about the Civitas developments over the last 6-7 years, and discuss some of the many undocumented claims regarding this product. Then we'll go into the experiments I did where we characterized response to the Civitas in the field, and then brought it down into the growth chamber to do some physiological assessments. So spray oils have been used in agronomy since the first published report was from 1787. And those were plant-derived oils. And then when we had the development of crude oil refinery distilling, there's an explosion of all these different byproducts of oil, and people were trying to figure out what they can do with them. And so in 1890, Cook summarized some work people have been doing, looking at putting kerosene on citrus and finding out that it had beneficial effects to prevent insect problems. And then it was very common in vineyards to actually go out with a 25% emulsion of crude oil and just put it on apple orchards and vineyards to help with disease pressure. We've come a long way since then. Those old compounds were really a big mixture of aromatics and unsaturated hydrocarbons that can be kind of nasty and toxic to plants and people. Now we've gone to these lower aromatic, very saturated compounds with narrow distillation temperatures, which is pretty much a paraffinic spray oil that we have today. Paraffin is just an older term for an alkane, if you're more used to that terminology. It's a fully saturated carbon molecule. Some of the consequences of spray oils, the first obvious advantages that we see: control of insect pests mainly, and now we're actually seeing there's some impact on pathogen pests. It's got a low environmental impact and low human toxicity. Some of the disadvantages, though, however, are they don't guarantee control. Sometimes you'll have breakthroughs, sometimes you won't have great control with the products, and there's always the potential for phytotoxicity, even with the most refined spray oils that we have today. So here's a picture of a conifer that was sprayed with a dormant application of a spray oil in the winter. It was sprayed too late. The temperature heated it up, and we had some strong phytotoxicity in this conifer. So it's common across all different crops that these phytotoxicity can occur. We also see that these hort oils are really good spray adjuvants. So mixing them with traditional herbicides, insecticides, or fungicides at lower rates, we see better control because these products help to get the pesticides into the plant, usually helping to get it through the cuticle. So here's an example of a turfgrass plot. Was surrounded by dollar spot, Sclerotinia homeocarpa, then mixed with half of the low labeled rate of a really low impact fungicide, and we're getting really good control of that dollar spot disease. But phytotoxicity can be a problem. And if you look at the literature, there's kind of two types of phytotoxicity. There's acute phytotoxicity, and that's direct damage to the leaf. So leaf lesions are common. If you have fruit, you have sunburn. But really what it comes down to is the membranes themselves are being damaged by these compounds. This is generally more true of the older chemistries that had a lot of aromatics in them and a lot of unsaturated, highly reactive hydrocarbons. Horticulture oils today generally classified as chronically phytotoxic. You see a decrease in growth, decrease in photosynthesis, transpiration, and respiration. And this mainly occurs because the oil persists on the leaves, and it blocks the stomates and inhibits gas exchange, and you have all kinds of feedbacks then that's going to suppress these processes. So now more generally about Civitas. It was developed in the mid-2000s. It's developed from food-grade isoparaffins, which they use to make everything from cosmetics and lubrication to equipment that's used for food processing. It was labeled as a fungicide in 2009. It also got OMRI certified by the Organic Materials Research Institute as a certified organic product, so that's great for its use. It has limited fungistatic activity, and the thing that makes Civitas different, at least in the literature, compared to the other horticulture oils, is it's been found to induce systemic resistance. So application of this product primes genes in the plant defense pathway, so that when a pathogen attacks, the plant is really easily, quickly to respond to that pathogen attack and suppress the disease. So why isn't everybody using this product? I mean, it seems like it's great. It's an OMRI certified product. It helps out with all kinds of myriads of disease issues and insect pests. So there's a lot of anecdotal claims that came out when the product was first released. The biggest one was golf course superintendents thought that their clipping yield on their putting greens had increased. And we know that an increase in clipping yield can be negative on a turfgrass health standpoint because it depletes storage carbohydrates that could be used to help recover from stress in the future. And then the other big thing for superintendents is it reduced ball roll distance. So here's a picture of a guy measuring ball roll. It's a pretty simple little device. It's just a stick. You lift it up. When it gets to 20 degrees, the ball rolls off, and then you see how far it rolls on the green. And although that may seem like a silly thing, that's really important to golfers. Golfers associate fast ball roll distances with really nice, high-quality facilities. So superintendents try to maximize ball roll and limit practices that might somehow hinder ball roll distance. So we wanted this look in the growth chambers to kind of see, is the grass actually growing faster? And we have a time lapse to kind of show that it appears that there is an increase in the leaf elongation with the Civitas treatment compared to the control. It's not super dramatic, but it is seem to be occurring, and we've also measured this in the field at putting green height turf with some time-lapse photography. Finally, the other reason people that this product isn't used that often or is struggling to gain acceptance is because it is phytotoxic to turf. And you can see in the top left corner there, the Civitas plot. It's a little bit, it's hard to see with this projector, but it's a little bit more brown, a little bit less healthy than the control. And so the company solves this by mixing in a green pigment to just paint the grass green. And so that really helps to minimize that problem. So at the onset, we had a lot of different questions, and it was really difficult to figure out how exactly we're going to proceed. I came here with all these different anecdotal claims, like a disco, and I needed to try to figure out some kind of way to link all these different responses into a dissertation. So kind of here's a roadmap. Now after I've gone through about probably 15 different experiments in the field, in the growth chamber, in the greenhouse, over the last 6 months, I've been finally able to take those experiments and try to make sense of all these different results. So we first quantified the response of Civitas in the field by measuring ball roll and clipping yield and surface characteristics like density and canopy temperature on a fairly regular basis. Then once we got a good sense of what's going on in the field, we were able to go into the growth chamber, look at phytotoxicity, and try to understand, is it direct damage for an acute phytotoxicity, or is it more of a chronic damage? Then we expounded upon that, looking at gas exchange, because most of these modern oils have been found to inhibit gas exchange and photosynthesis. And then finally, if we're inhibiting gas exchange, what does that do to the carbon dynamics of turfgrass plant? So in my second chapter of my dissertation, we're quantifying just the clipping yield and the ball roll distance. And that was the main objective. It's just kind of look at clipping yield and ball roll for 10 days after we applied the product. We measured it every single day. And then we did that 6 times over the course of 2 seasons. Then we also wanted to look at some kind of mitigation strategy if there is an increase in ball roll, so that this product can gain acceptance in the field. Our hypothesis was that ball roll decrease that was seen in the field was inversely related to the increase in clipping yield with Civitas. And we were hoping that the plant growth regulator trinexapac ethyl, which is commonly used to inhibit cell elongation, might actually mitigate that decline. So all these field experiments, I'll just kind of set them up here. They're done at bluegrass lane on a bent grass, annual bluegrass putting green. They were maintained to standard practices. So in the clipping yield response study, again, we had 6 10-day runs. We collected clippings every day. And we had to evaluate the different components of Civitas individually to try to get a better sense of what's going on. So we have a control. We have the Civitas oil by itself, the harmonizer oil, pigment by itself, then the mix of the two, which the company and is referred to in industry as the 2-pack because you mix it on site. And then recently the company, Petro-Canada, developed Civitas 1, which is mixed at the factory. It has a different emulsifier. So it does have a little bit different properties, which we'll see. So the ball roll measurement then kind of mirrored the clipping yields. It's just done in a separate putting green. And we looked at ball roll distance over the course of a day and then across 6 10-day runs. And we eliminated the treatments here to just the control, the 2-pack, the 1-pack, the Civitas 1, and then with and without this growth regulator trinexapac ethyl, which reduces plant growth. So here's kind of a look at what it looks like in the field. The Civitas, and then the 2-pack, mixing the two together, and then the control, and then the harmonizer by itself. For the clipping yield, we found that every time we applied oil, we had an increase in clipping yield. Whereas the harmonizer did increase clipping yield, and the control obviously is the control. There's 2 hypotheses here. First, it's growing faster, and that's why there's more clipping yield. And that's kind of supported by our time-lapse measurements. The other hypothesis, though, is that the oil itself may be contributing to the weight of the leaves. We're only talking about milligrams of tissue difference here. And so that's definitely a possibility. We think it's probably some combination of both of them. Some of the oil has gotten down into the canopy, into the verdure, and so not all of it's stuck on where the grass actually being clipped. But it's hard to actually just say that the clipping yield increase is only an effect of increased growth rate. So then we looked at ball roll distance, and we saw that, in fact, the ball roll distances did decline with the Civitas 2-pack and even more with the Civitas 1-pack. These are practically significant differences and statistically significant differences. So this is at a level that may not be acceptable to golf course superintendents and the golfing public. It lasted for all 10 days. We see an increase in ball roll as each run progressed because the plots were rolled, and so as we were rolling it every single day, we increased surface uniformity and the ball would roll farther. Unfortunately, there's no interaction with the plant growth regulator, and the growth regulator only increased ball roll by about 7 centimeters, which isn't even significant or practically significant in the field. Golfers can't perceive that small of an increase. So the growth regulator didn't help out, although it did counter the growth increase with the Civitas. So if you look at this data, and you look at the clipping yield data from the previous slide, you would then assume that as you had more clipping yield, you're going to have slower ball roll distances. So we did a correlation to see if that is in fact the case. We found out it's not true at all. This has been combined across all different days, but it was also done for each day, each measurement within a day, on each run. And there's only like a handful of occasions where there was a correlation of clipping yield and ball roll distance. And so this is important because the literature suggests that ball roll distance is correlated with clipping yield, but they never actually do correlations. They look at clipping yield, and then they use a product that looks at, they'll put fertilizer down. They'll see the grass grows faster. They'll see the ball roll goes down, and then they just assume that, oh, the ball roll went down because the grass grew faster. But they never actually do the correlations to prove that. And so I think there's some work that could be done there to better understand this, because there's got to be some other factors for leaf elongation that's influencing ball roll. If it's the leaf friction, the firmness of the surface, something we need to figure out more. And so it's so essential to management of golf courses. Quick conclusions. Civitas increased clipping yield, which may have potential implications for reducing carbohydrate levels in the plant. Ball roll increased. Civitas did reduce the ball roll, which could be a problem for high-end putting surfaces, and their plant growth regulators just didn't have enough of an effect to really cancel out that decline in ball roll distance with Civitas. So then we looked at the chronic phytotoxicity with Civitas by first characterizing it in the field and then isolating the cause in the growth chamber. And we hypothesized, due to the nature of this oil, that Civitas will reduce gas exchange, which is going to cause some chronic phytotoxicity, and that the harmonizer masks this effect in the field. So what we did is we had another study at bluegrass land on the same putting greens as described previously, and we had a factorial, 2x2 factorial. We have Civitas, the control in Civitas, and then the oil by itself, and then the oil and the Civitas, the pigment by itself, and then the oil and the pigment together. And did turfgrass quality rating, which just kind of looks at the uniformity of the surface, the color, the density. It's very much subjective, but it's also very repeatable at the same time. We looked at chlorophyll index with the CM1000 to get a sense of the amount of chlorophyll in the leaves, and canopy temperature with a FLIR camera, and then at the end of the study we did tiller density counts. So here's kind of a picture of what this phytotoxicity looks like at the end of this growing season. You'll notice the control, it looks a little thin. It's at the end of the growing season. It's been warm. The POA annual bluegrass is starting to be stressed out. Put the harmonizer on, that pigmentation really helps the surface look a lot better. And I also suspect that it actually helps the plant to cope with some of the light stress during the summertime. The Civitas then, you had the typical chlorotic appearance, some general thinning going on, so starting to have a decrease in quality. And the 2-pack, again, had that same thinning, but then it was still painted green with this harmonizer. And so where the grass did die, it was very obvious by these little flecks of blue. So we looked more specifically at how it changed over the course of the year. So we looked at canopy temperature with a FLIR camera, and you can clearly see where the control plots are versus the pigment and the oil and the mixture of the two. We did find that there's an increase in temperature with the oil and the pigment, which you may expect. And it was definitely a function of solar radiation. So when it's sunnier out, there's larger differences. If it was cloudy or raining or cool, generally there was no differences between the treatments. And although this may seem significant, the canopy temperature increase is only on average a half a degree warmer with Civitas and 1 degree warmer with Civitas plus the pigment. And even on the sunniest, hottest day in the summer, that increase was only 2 degrees C. So it really may not be that and physiologically important to have that small increase. But it does suggest there may be something going on, like reduced transpiration with the oil. Finally, the phytotoxicity with the quality. This bottom line here with this dash red is the Civitas, and you can see in the first year it was below the acceptable level of 6 for a majority of the summer of the year, whereas the pigments Civitas, the solid red line, was at or above the level of the control the entire time. The interesting thing in 2013 was the 2-pack declined at the end of the year to a level that was unacceptable, and you saw that in the last picture with the thinning of the grass. So the hypothesis is this harmonizer is actually just more masking this phytotoxicity, because in 2012 we didn't have the thinning, we just had the chlorosis, which was masked by the harmonizer. But then in 2013 we had the chlorosis and then the eventual decline in density, which then obviously can't be masked by a green pigment. Yep.
How often were these applied? Is that once or?
They were applied, every 2 weeks.
Every 2 weeks.
Yep. At the end of the study, too, we did density counts, and we found that we had a definite decline in stand density, number of tillers, with the Civitas and the 2-pack treatment compared to the control and the harmonizer. So there's definitely some definite phytotoxicity happening here. And we wanted to figure out, why is this happening? What causes this phytotoxicity? With some implications of reduced gas exchange with an increase in temperature, then how would that affect both synthesis and respiration, and how would that affect carbohydrate pools. So we then went into the growth chambers in Wow Hall for the rest of my experiments I'll be discussing, just to kind of get it set up. All the experiments happened in the same environmental conditions unless they were otherwise noted. 30 degrees C during the day, 20 degrees C at night, about 500 micromoles of photosynthetic reactive radiation per meter squared per second for 16 hours. And then we established these cones of annual bluegrass for 3 months, and once they were dense and looking like this, then the experiments happened and they were fertilized weekly with a low rate of nitrogen. So understanding phytotoxicity is a bunch of small little experiments, so we'll just kind of go through these quickly. We looked first at membrane disruption because that's a sign of acute phytotoxicity. And to measure that, we just looked at electrolyte leakage. And we found that there was no treatment differences relative to the control, which is good, showing that in fact these Civitas isn't acutely phytotoxic. It doesn't damage the plant in any way. Interestingly, there was an increase or decrease in electrolyte leakage with the pigment, although all the values are really minimal, which suggests that the plants are healthy. And I believe we can explain that because when you measure electrolyte leakage, you measure the electrical conductivity of water that's got tissues floating in it for 24 hours. Then you autoclave that tissue and you measure it again to see what the potential amount of electrical conductivity can be. And I think what happens is the pigment with this huge chelated copper compound, it probably drops that copper out, so then it increases the denominator of the equation and dilutes the electrolyte leakage number just because you're losing the chelation of that copper ion. Next, we went to look at scanning electron microscopy of some turfgrass leaves treated with Civitas 24 hours after application, and then again 48 hours after application after a second application of the product. And we find that there's some definite signs of change in the morphology of the cuticle, where we have these nice platelets in the control that degrades down and becomes more flat. And we see oil persisting on the stomata, which may affect gas exchange and actually may be beneficial to pesticide absorption. So we see that the cuticle is being modified in morphology. We wanted to verify that it wasn't changing in quantity or composition, so we extracted the cuticle off of leaves treated with the 5 different formulations of or components of Civitas, and we found in the top total ion chromatograph, these represent a lot of the waxy components. So you have some fatty acids, and then these are all primary alcohols ranging from 22 to 28 carbons. The predominant one was a C26 primary alcohol, hexacosanol, and that level didn't change with our response to the Civitas. There was, however, a really pronounced Civitas signature that was found on all of the total ion chromatograms from the GCMS. And when we look at it from a mole to mole ratio of the total moles of Civitas versus the total mole of the 1-hexacosanol, we find that there's between 9 and 17 times the amount of waxy oil substance on these cuticles than there would be if they were just if they're natural and not treated. Finally, we did some gas exchange measurements with a Lycor chamber. It's got an Arabidopsis setup, so we can drop our cone right in there and adjust the light intensity and make light response curves and ACI curves really simply to look at photosynthesis and respiration. And what we found was that Civitas reduced radiation use efficiencies. The Civitas 1, the blue line, was more injurious than the Civitas 2-pack or Civitas by itself, and the pigment had no effect. This is only done 24 hours after application, and so we repeated this experiment to get a better sense of how gas change is being affected. But just looking at phytotoxicity, we determined that acute phytotoxicity did not occur, and the waxy epicuticle wax composition and content was unchanged. But then there were some signs of chronic phytotoxicity resulting from that persistence of the oil, which is inhibiting gas exchange. But more research was required to figure out how long does that actually last, how bad is it. So we designed the next chapter of a dissertation to look at how Civitas reduces gas exchange. And so this was a lot higher, larger level of measurements. So these gas exchange systems are really great because they're super powerful, and you can make carbon assimilation or transpiration response curves to factors such as photon flux gradient, which is Q, or ambient CO2 or internal CO2, Ca and Ci. Because I'm doing canopy level things, I have to do Ca. I can't actually calculate Ci because of the, you can't measure an individual leaf of turfgrass. You can also look at under different vapor pressure gradients. And then from those models, we can estimate things like quantum yield, rubisco activity, and respiration. So how do these devices actually work? Here's just kind of a graph of a light response curve that I did. I made my stability be kind of quick so I could fit it in the course of 10 minutes. But what happens here on the x-axis, you have the time, and then photosynthesis and transpiration on the y-axis. And so the cone gets put in, it's at 500 par, and then once that stabilizes, the machine automatically increases light intensity to 2,000, and then it responds. And once it comes back to a stable level, which I had defined, and then here it wasn't entirely stable for this example, then the machine proceeds to reduce the light levels, or in the case of a CI curve, reduce the level of carbon dioxide in the chamber to measure photosynthesis. And then we can plot all those points where it makes a measurement, and we get these great light response curves that tell us a lot of interesting things. The y-intercept talks about the maximum rate of respiration. Light compensation point. That initial slope represents quantum yield, or the amount of carbon dioxide molecules fixed per photon. Then the plateau represents the maximum photosynthetic capacity of that plant. And that decay is a result of either gas diffusion inhibition or RUBP regeneration limitations. And in the previous experiment where we showed the light response curves, we just did a simple ANOVA. In these experiments, we used regression analysis. And so we were able to then do regression and solve for respiration, the Amax term, and the apparent quantum efficiencies. The other great thing, too, is we can look at the transpiration rate. And because transpiration is a factor of leaf conductance times the vapor pressure deficit, which we can fix with the LICOR, we can assume that transpiration rate and leaf conductance are a good proxy for each other. They're pretty proportional. And that can get to our questions about stomatal inhibition. So we did one experiment where we put a cone in the LICOR and then made an AQ curve at different light intensities, and then right after that it went to an ACA curve at different levels of carbon dioxide with a constant relative humidity and light level for the carbon dioxide, so we could have one vapor pressure deficit value to then make those comparisons on leaf conductance. And we measured this on the control, the Civitas 1 and the Civitas 3 hours, 1, 2, 5, and 10 days after we apply the products. And so what we do is we get these big time courses where we have 3 hours to 10 days, and then we replicate our runs twice. And so you'll notice that for all these treatments, there was never a difference in respiration. So the Civitas didn't affect respiration, but it did affect the Amax levels, which then also means it affects the quantum yield of the plant. And in run 1, we had more suppression of Amax than in run 2, and that was a factor of a misapplication and a little bit higher rate because the nozzle was too close to the canopy leaves. And so it got a little bit higher rate, and so it definitely showed that there's a rate response, and I've done some experiments to show there is a linear rate response with Amax values. But kind of just to summarize, what this shows is we definitely have a decrease in radiation use efficiency, and respiration wasn't affected. And it takes 10 days at least for this effect to start to wear off. Transpiration from that same study, again, we have photon flux density on the bottom. There was no interaction. So as we had brighter light intensity, we saw an increase in transpiration, which makes sense from a mass balance standpoint. But the Civitas had higher levels of, or had lower levels, lower intercepts, showing that there's less transpiration. And it wasn't as dramatic of a decline as the light response measurements, the simulation measurements. And usually recover within 48 hours. By 48 hours, you can see that the transpiration rates came back to the level of the control, which is interesting. And then finally, we looked at the ACA curves. And this kind of looks at the kinetics of rubisco activity and carboxylation efficiency of the plant. So the initial slope represents the initial carboxylation efficiency of the plant. And then the y-intercepts represent the respiration rate. And you'll notice, first of all, respiration rate was different. It's a little bit lower with the Civitas treatment. And that results from an increase in photorespiration because these are all at the same light intensity of 400 or 500 micromoles of power. And so this data kind of indicates that there's reduced carboxylation efficiency and increase in photorespiration. And we know that when you have a decrease in gas exchange, that activity of rubisco declines because it takes a lot of energy to keep rubisco in an active state, especially if it's limited for its substrate. And these curves suggest that the plants are substrate carbon dioxide limited, and therefore they have a decline in rubisco activity. And then finally, we looked at gas exchange, carbon assimilation as a function of transpiration rate, which is a proxy again for gas exchange and leaf conductance. And we find with the Civitas 1 that there is a really strong increase in photosynthesis back to kind of level of the control as we had higher transpiration rates. So this suggests directly that stomatal conductance is actually what's inhibiting this carbon assimilation rate. The Civitas itself was a little bit less, which makes sense because that's reflected back on the light response assimilation curves. Another thing to note too is after this initial part where it's really linear or fairly linear, that occurred early after we applied, and then later on after we applied the Civitas, it came back to be less of a function of transpiration rate. So that suggests that it's an inability of the plant to utilize all the carbon it's taking in from transpiration, from gas exchange, again suggesting that rubisco is probably somehow inhibited through some kind of inactivation later on into each run. So the conclusions, Civitas definitely reduced carbon radiation use efficiencies in transpiration, gas exchange for a period of 10 days. And the carboxylation efficiencies were definitely reduced as a function of that decline in gas exchange measurement. So then it leads us to our final questions is, so okay, now we know there's less photosynthesis occurring, less assimilation of carbon. What's happening to the partitioning of carbon? And then another question would be, what does this reduction in gas exchange have effect on oxidative stress in the plant? So the final chapter of my dissertation, we looked at Civitas altering carbon dynamics. We measured plant biomass partitioning, and then measured concentration of storage carbohydrates. This experiment was designed to simulate stressful conditions, so it's under really high light intensities. And that partly was because we find that as light intensity increases, there's a bigger disparity between the Civitas level and the control for carbon assimilation, so that way we kind of exacerbate it. It's definitely a worst case scenario. Additionally, it led to some episodic daily water stress. So we kind of could evaluate how Civitas affects plant stress tolerance, which is something that's marketed a lot that is useful in helping plant health. So we reapplied Civitas and wilt stop, a commercial anti-transpirant, bi-weekly, and then measured carbon partitioning and quality and gas exchange over the course of 8 weeks to get a sense of how carbon partitioning is changing. So first, we did confirm that the Civitas and the wilt stop reduced carbon assimilation and transpiration by about 10 to 15%. And this was measured 2 weeks after we applied. So there's even lasting inhibition of carbon assimilation 2 weeks after application. When we look at it, there wasn't an interaction with weeks after treatment, but we did find that in the first 4 weeks, there's kind of a general increase in carbon assimilation and transpiration. And I think that is a function of the plants adjusting to the new higher light intensities. However, the episodic drought stress that occurred started to really deteriorate plant health. And at week 6 and at week 8, there's a dramatic decline in carbon assimilation and transpiration rate due to that high stress environment. So how did it change biomass? Clipping yield was the only thing that was significantly different in biomass. With Civitas, Civitas had higher clipping yield again. Again, the time lapse would suggest that it's actually that there is some growth component to that, but it could also be the weight of the oil. And counter to that, though, wilt stop does have weight itself, so then you'd also expect to see an increase in wilt stop, and that didn't occur. There's no change in the above-ground biomass or the root mass, although there was a trend of decreased root mass with Civitas. And the water contents were the same across the three. It's also obvious, too, there's no interaction between treatments and weeks after treatment. But on week 8, we start to see there's a dramatic change in the mass of clipping yield, and especially the root to shoot ratio. You know, at week 2, there's about double the amount of verdure as there is to their roots, and at week 8, there's nearly 70% the amount of roots than there are above ground tissue. So that drought stress is definitely changing how carbon's being partitioned in the plant, and we see a decline in water content as a result of that drought stress. So how does Civitas affect storage? Carbohydrates. When we put Civitas down, it reduced the starch in water-soluble fructans in the turfgrass. So we think about water-soluble fructans as turfgrass's currency. And if you're doing any practice that's reducing the amount of sugars in the plant, then you're potentially reducing the plant's ability to recover from stress later on during the year. Interestingly, for the weeks after treatment, the starch levels decline with drought stress, but we see an increase in water-soluble carbohydrates as the stress continues on over the course of 8 weeks, which some of the literature suggests would be consistent with the literature and suggests that it's the plant then preparing for regrowth once that stress ends. And so finally, we looked at soluble sugar response to these episodic drought stress. And so these are the main sugars that were found in the above ground biomass of our turfgrass. We had fructose, glucose, and sucrose, myo-inositol, sorbitol, and trehalose. There was no interaction with treatments for myo-inositol, so they were all pooled together. And we noticed some interesting trends. First, looking at 2 weeks to 4 weeks after application, you notice that the treatments were all statistically the same. And then as that drought stress, episodic stress really picked up, then we had a real divergence in the level of soluble carbohydrates in the tissue. And the plant uses soluble carbohydrates for osmoregulation, and it also can use them for signaling of plant stress. So if we focus just on the 8-week measurements, we notice that for fructose and glucose, we see an increase in those sugars with the Civitas compared to the control, and the wilt stop was in between. However, for the sucrose, there wasn't an increase between the Civitas and the control plot. So the control really had a dramatic increase in sucrose, which the turfgrass literature and Kentucky bluegrass suggests sucrose should increase during drought stress to help it with osmoregulation. Then look at sorbitol and trehalose, some stress sugars. We find, especially with the trehalose, that there was not a response to drought stress with trehalose content, whereas the control did increase under that drought stress. So it brings up some questions. They're really unclear currently, and it's going to require a lot more experimentation to understand how Civitas is actually affecting plant stress tolerance. But from this, there is potential that maybe the Civitas is actually hindering normal plant stress, stress response. And if you look at the quality data, which I didn't present, you would notice that the Civitas probably handled the drought stress worse than the wilt stop or the control did. So there is some potential that this could be a negative response, that the plant is just not perceiving the drought with the Civitas application. So kind of in conclusions, we see the increased clipping yield in Civitas at the expense of carbohydrate accumulation, which has implications for stress tolerance. And going back to the beginning of the study, that could relate to that decline in turfgrass stand density by the end of 2013. We did see the obvious improvement in did not see an obvious improvement with turfgrass stress tolerance with Civitas, which is kind of contrary to what their marketing campaign would suggest. And we're still unclear why soluble sugars responded the way they did, and it will require some future experimentation. So to kind of look full circle, what's happened, we've shown that Civitas affects clipping yield and ball roll, which may limit its application to putting green, but still has potential for fairways and roughs and tee boxes and athletic fields and other places that want to reduce their pesticide usage. And there was this phytotoxicity that results from this product. And it's definitely a chronic phytotoxicity from stomatal inhibition, which reduced photosynthesis and transpiration. And the carbon dynamics indicate that we have a decrease in soluble or in storage carbohydrates with the Civitas. So some future experimentation would be look to optimize the dose response. Everything I did here was a worst case scenario. If I go at less of a rate, we'll have less of effect on physiology, but then counter to that, will it have a less of effect on the disease priming aspect that's the product is used for? We should also measure oxidative stress. If we're inhibiting gas exchange, it's easy to believe that there's some difference in photorespiration and photoinhibition. We should look at the formulation of the product. Right now, it's a pretty wide, they're all paraffins, but they range from 16 carbons to 33 carbons. Generally, products that are less phytotoxic, like the JMS stylet oil, they have really, really narrow ranges of paraffin chain length. And so potentially, maybe there are components of Civitas that are phytotoxic and they're not increasing the plant defense. Specifically, I saw a paper recently, got published like a couple weeks ago, that showed a 16 carbon alkane has been found to prime induced systemic resistance and has a lot of potential to be the active ingredient of Civitas. And so we should look at that formulation more closely. And then finally, I'm trying to understand how Civitas affects stress tolerance, because this product is being applied during the stress of the summer season, and we should get a better sense of how that is actually occurring. So I'd like to thank a lot of people, especially my committee, Frank, and then Dr. Setter, Bergstrom, Mazurek, and Marty Petrovic. I thank my research techs, Mary Thurn, Brett Welch, and my undergrads, Jordan Gary, and Angelo Fever, who tirelessly counted, did a lot of things like ball roll measurements every day, and cleaning grass clippings and counting tillers. It's not fun work. I'd like to thank Petro-Canada because they funded my Ph.D., and also Michael Pfeffer, who is a lead scientist that developed the product. He's very, very helpful to work with. I could thank Bill Miller for helping with the carbohydrate analysis, especially, and Rose Harmon for actually helping me do the analysis twice. So I could thank Liling Chang and Tom Whitlow for letting me use their Lycor, and the Wild Hall plant care people, especially Nick Van Eck, who was very, very helpful. I'd also like to thank my fiance, Katie, because aside from her helping me do a lot of lab analysis on weekends, she's always very, very supportive. You all know Katie, and she's a sweet, caring, compassionate person. And then all my friends that have helped me survive these 3.5 years at Cornell, and helped me survive at the Big Red Barn. So with that, I'll take any questions, and thank you very much.
This has been a production of Cornell University, on the web at cornell.edu.